Ladies and gentlemen, please welcome to the stage a representative from the Zuckerberg Initiative.
Okay, very good. Good afternoon. My name is Jonah. I am the Vice President of Scientific Strategy and Partnerships at the Zuckerberg Initiative. And it's really a pleasure. I'm excited to share with you all how we are both thinking about some of our specific AI initiatives and specifically our work in virtual cell modeling, but also one of the big challenges that in the biosciences, as well as AI more generally, that we face, which is a bottleneck around data. And associated with this, myself and my team and others across CZI have worked within biological data for a number of years. But in response to and thinking about this strategy, are also beginning to evaluate and start to generate some projects. And really what I hope is deliver these early data sets, but perhaps as importantly, is start to innovate on the models and the efforts by which large-scale data generation is undertaken. And importantly, that these are efforts that are done across the scientific community and really demand close partnerships between the private sector and the public space. And so for those of you that stick around for the whole talk, hopefully you can take some kind of specific examples of this that I'll share with you, and via this one project that we've initiated and are about a quarter of the way through, called the Billion Cells Project. But for those of you that have to depart a little bit early or wander around, really what I hope to convey is this delivery of data sets, but also the innovation of new models to drive forward progress in AI. So let me first just very briefly introduce everyone to the Zuckerberg Initiative and our mission. So the Zuckerberg Initiative was founded now nearly 10 years ago by Priscilla Chan, as well, and with her husband, Mark Zuckerberg. And the organization is really oriented towards solving problems, and specifically problems in fundamental research at the intersection of science and technology. And this is all directed towards a mission statement that is to create technology that reveals the inner workings of human biology and accelerates progress, and that acceleration then in turn makes it possible to cure, prevent, or manage all disease by the end of this century. So this is something that we believe very strongly in and have been working on for a number of years. Again, I would point both to Mark Zuckerberg's experience as a software engineer, as well as Priscilla Chan's expertise as a pediatrician, that in many ways CZI is the confluence of their expertise of bringing science and technology to fundamental research questions, such that we don't understand one disease at a time, but start to develop tools and technologies, of which AI is a perfect example of, that can accelerate all of our progress towards really any disease that we currently know about, as well as others that may emerge in the years or decades to come. And currently at CZI, where we're focusing on within that large realm of life sciences and biomedical research is the unit of the cell. And for biologists, this might intuitively make a lot of sense. For those of you that have not thought about biology as much, I would frame it this way. One, for the last number of decades, we've been really focused on molecules, proteins, genomes, transcriptomes. And by moving into an order of cells, we're starting to think about how those molecules begin to holistically come together and both regulate homeostasis, but also respond to disease. The other framing, and I'm a cell biologist and geneticist by training, is that cells are the fundamental unit of life. And also, when we think about genetics and predispositions to disease, many diseases we now know have a genetic predisposition, but all diseases have a cellular mechanism. And so by understanding this fundamental unit and understanding it holistically and being able to model aspects, both again of homeostasis and also response in disease, the unit of the cell is an incredible opportunity to generate new data sets and also drive forward in these rich representations of biology that we believe, as well as many others, will be fundamental to understanding human health, disease in a very holistic and cross-cutting sense. So back to this mission of being able to create science and technology to support and enable the prevention, cures, and treatments of all disease. Okay, so when we think about that goal of holistic cell modeling, and when we look around and begin to try to orient our work and the rest of the world's work in AI models towards these holistic visions of cells, we all of a sudden hit what I find to be a fascinating and wonderful paradox within biology right now. And that is that if you go and you talk to any individual researcher, it is very likely that you will hear a series of challenges that they face in terms of the amount of data that they are facing and routinely have to work with and utilize, whether it be imaging data, sequencing data, mass spec data. The ability to generate data is vast. And similarly, if you go look in archives and repositories, whether they be NCBI or GEO or others, the volume of data that is there to be utilized is, again, significant, perhaps greater than the Library of Congress, depending on how you tokenize and think about that data. And yet, if you then turn around and go have conversations with researchers that are interested in generating new foundation models, what you will very quickly hear is that we are data deficient, that we need data desperately. And so what I would like to put forward, and again impress on this group, is that while biological data is abundant, there is a clear and critical gap in the right data. And by right data, I would like to define it in a couple different ways. One is data sets need to be curated and interoperable. So large volumes of data are helpful and good, but what we really need are large volumes of data that have been consistently generated and annotated by the scientific community with labels and annotations and harmonizations that reflect the underlying biology, but also can then be easily consumed and utilized by those interested in training large foundation models or other models. The other thing that I would say about the data deficit is we need to begin to figure out how to generate these large interoperable data sets faster and at less cost. So currently the data generation process for many scientists operates on the orders of years, and I think as anyone walking around the Expo floor today would very quickly agree with, the pace of the modeling and the computational progress is happening on the order of days or weeks or at most months, and we have to bring those timelines closer together. So, more data, richer data reflecting the biology, but cheaper, more iterative, faster, and with greater interoperability. And so our approach to this is couplefold. One is as I mentioned, we're thinking about cells as units, but even within that larger vision and goal of modeling whole cells, we're also trying to take a fairly principled and focused biological focus. And here, what we're thinking about are different domains and questions where we know that there are incredible opportunities to train AI models, but also ones in which there is a large need and opportunity to generate these data sets, again, interoperable, fast, relatively inexpensive, but in a way that are directed towards questions that we believe that those data sets will begin to make a footprint in a field that then others can begin to contribute to with those same standards and senses of interoperability. So you can think about these data sets both as a way to begin to train models, but I hope also as a way to catalyze progress and knit together a community of international scientists. And so to give you a few examples of the types of questions that we're thinking about, I've listed 4, and these are kind of areas that we're really thinking deeply about within our virtual cell work. One is genome function. So we know from the human genome, from many other genome projects that have followed, whether it be cancer genomes or genomes from diverse populations across the world, that as a linear string, the genome has a lot of variability. We also know that the genome serves as this blueprint for downstream transcription, for translation of proteins, and ultimately has in it an encoding of how phenotypes and organisms develop. What we don't often understand are the rules and the logic by which that knowledge and that information transfer happens. And so we really want to understand how genotype to phenotype interactions are happening, and this is a wonderful space for models. And those phenotypes here can be quite diverse, right? So we often, as I mentioned, think about phenotypes in the disease context, but I'd also encourage everyone to think about phenotypes in the biological context, whether that phenotype be the transcription and kind of functional expression of a genome in a given cell and why they're all different, to protein diversity and other features. The next are cell types. So cell types within any one of our bodies, we have on the order of 35 to 40 trillion cells. all performing these exquisite and unique physiological functions. There's some redundancy in those cells, there's some exquisite functionality and specialization in those cells, and yet we really don't understand the nature of all of those cells, and in particular the interactions of them. And so we really are trying to utilize some data sets that I'll tell you a little bit more about in the next slide, as well as new data sets within the Billion Cells project that I'll tell you even more about, to understand how cell type diversity, again, in health and disease is being regulated. And then finally, two additional areas. One is gene function. So if you go and talk to many geneticists, we now know through the Human Genome Project and many other efforts of kind of the nature of many genes, and yet the function of those genes, and specifically to connect the two first bullets, the function of those genes in a cell type specific manner are very, very poorly understood. And the dimensionality of this question is also vast. If you want to understand not just the function of every gene, but the function of every gene in every cell type, and do so in an individualized way, so across genotypes, the space very quickly expands beyond really tractability. So again, a perfect place to think about generative models and also starting to learn. So let me now go back in time and tell you a little bit about some of the efforts that we've done historically. This effort in particular is one that I'm very proud of, and my team has been working on worked on really for the first 5 or 6 years of CZI's history, and is a tool that we called Cell by Gene. Cell by Gene is really a data science tool, here being applied to single cell transcriptomics data. And so for those that are not as familiar with transcriptomics data, this is a technology that allows you to isolate a single cell, human cells, mouse cells, whatever cells you're interested in, and then from that cell, measure all of the transcriptomes and the genes expressed in that cell. And so what you get out is, for a given cell, a set of features equivalent to roughly the number of genes in the genome. In humans, that's on the order of 20 to 30, depending on how you count. And so the impact is you get this very, very high dimensional view of biology. So across that high dimensional 30,000 feature space, you can then start to measure many, many, many cells and understand both the genes, but also that high dimensional embedding and representation. And so what we did in Cell by Gene is we enabled researchers around the world to contribute their data sets. We also funded a number of early efforts to generate these data sets. And now in total, there's 110-ish million cells worth of data, free, open to the world to use. But importantly, they're all curated. They're all conforming to a standardized schema. And they're all available both to explore at the data set level, but also at the single cell level. So if some researcher came in and said, I really care about T cells, and I'm interested in how T cells across tissues or studies are behaving, or the molecular signatures of them, because I want to develop a new immuno-oncology product, you can go to Cell by Gene and actually segment and pull out all of the T cells, only the T cells, and kind of get rid of the other tens of millions of cells worth of data. And then what you have is this wonderful resource that can quickly be summarized. And I will say that this is an area, and this resource is one that many researchers now around the world, on the order of about 10,000 a week, are using. And a good number of those are now starting to train foundation models on this data because of that high dimensional view of biology that the data sets offer. So that's a wonderful success story of how these early research can both translate into various types of AI models. The downside here is, when I was introducing Cell by Gene, I mentioned that it took on the order of years to build. It's about 100 million cells worth of data. I also told you that in one of our bodies, there's on the order of tens of trillions of cells. So we're talking about an incredibly small representation. It also took on the order of tens to hundreds of millions of dollars to generate. So this is a wonderful resource, and in some ways it has parallels to another incredible resource, which this audience might be familiar with, which is the Protein Data Bank. And this is a resource that I would say is really kind of the gold standard of community resources that focus on protein structure, and has been a catalyst for many efforts around the world where researchers can generate these structures that are very, very labor intensive, very expensive, contribute them in a standardized way and have them QC'd, and has been the substrate for many models that look to predict protein structure, including AlphaFold, which won the Nobel Prize last year. So what you see here are two examples, and the examples are these wonderful resources that are biologically rich. They're providing a substrate for AI models in biology right now. What they also are efforts that took a long number of years to generate, hundreds of millions to billions of dollars to generate, and both provide a proof point, but also point out some issues where if we want to generate more resources like this, we clearly need to think about how to generate an order of magnitude more data for an order of magnitude less cost. And yet I would say a success point here is also harnessing the scientific expertise around the world and the knowledge that scientists have to kind of bring those annotations and those data sets to these community-generated resources. And so kind of going back to Cell by Gene and how my team built this a number of years ago, we largely worked with a network of labs, about 500 around the world. Increasingly now, many more labs are contributing their data sets in such a standardized manner. But the way that this largely worked was that individual labs were given grants, and that with those grants, they would go through and do the very difficult and kind of specific work to collect cells, capture those cells, generate these libraries of the transcriptomes that I very briefly described, and then make some individual decisions in terms of sequencing and how to process those cells that were very, very nascent. We then also expended quite a bit of resources and time to avoid each one of those data sets going to a different database or existing in that kind of large amount of data, but low amount of interoperability and curation. And that was really the work that Cell by Gene provided that I gave you that example. And so the success there is over those years and through this model, we were able to collect and curate and now allow and enable access to hundreds of millions of cells. But this, again, took on the order of years for any one of these projects, and also leaves both quite a bit of biological and technical variation, which provides some challenges for the models. So, as I kind of briefly touched on, many groups are utilizing the Cell by Gene data resource to train AI models, but I would be the first to admit that there are some challenges with that in terms of differences in how the data were processed or normalized or questions like that, that would help the technical features, but were really kind of fit to the specific biological questions. And so what we have begun to think about now are new models of data generation that try to address some of these challenges. And again, the specific challenges that I've alluded to are greater pace of data generation such that it can keep up with and stay in line with the model progress, reducing costs, and also increasing standardization and interoperability while still enabling as much biological rigor and value in that data as possible. And so the Billion Cells Project is exactly that. So it is an initiative that we have launched just earlier this year in February to generate 1 billion cells worth of data. So again, an order of magnitude more than is currently available in Cell by Gene in the next 2 years. So this is a project that we anticipate having done by 2026. We're already close to 100 million cells worth of data that have been generated, and it's now really just starting to ramp up. And the way that we're doing this, again, is through deep partnership with the scientific community, but also with a number of different companies that are helping us kind of scale both in terms of efficiencies and also tapping into economies of scale. And so what this looks like is for those areas that I articulated, genome function, cell types, gene function, we're working with individual labs that are experts in collecting those samples or understanding the biology that, frankly, in most cases are simply capped because of access to the reagents and the sequencing costs. So they cannot operate at that scale, even though they can generate and capture those cells that the world is eager in. And so the way we're removing that bottleneck and enabling them to get deeper into the biology that they care about is by working with reagent providers, and specifically here within the single-cell genomic space, it's 10x Genomics and Scale Biosciences. But you could easily imagine as we move into new modalities, whether it be imaging, mass spec, or different domains, that new partnerships will flourish. And then as those cells are captured in a standardized way, all of that data gets processed at the sequencing level, as well as the standardized processing level in central pipelines that our team is providing to those labs and also engineering. And the net result is that all of the data flows through these pipelines with standardization. It is all deposited into a central database whereby the labs can have access to it, as well as our team and hopefully many other teams in the future can start to access not just individual data sets, but also begin to learn across all of these data sets. And this is happening instead of on the order of years, it's happening on the order of weeks or months. Some, again, variation mostly on how difficult the cells are to collect. You can imagine that if you're working in disease samples and human patients, there's going to be some different considerations than if you're thinking about drug treatment in mice, for example. And so this model, I think, is one that is robust, and I'll tell you a little bit more about some of the projects, but would also like to kind of leave people with the idea of thinking creatively about how to avoid simply creating more data silos by just large, very inward-looking data generation, but utilizing and in many ways kind of unharnessing the scientific community to generate data sets, but generate them kind of through the lens of modeling efforts in a much faster and much more iterative and rapid way. And so early on, these partnerships, as you'll see, kind of span, again, both private and public partnership. They include some of the sequencing and single-cell capture companies that I mentioned on the prior slide, but also leading research institutions, whether it be Yale, UCSF, Stanford. We're really curious not just about cell type function, but also how cell type function has evolved over evolution. And evolution, for those that follow the protein space, has been a really important factor in understanding function. And so organizations like the Marine Biological Laboratory and University of Chicago are great examples where they have incredible resources for organizations. Okay, so what comes next for us? We are obviously pushing really hard to get through these milestones, and the billion cells in 2 years is something that when first talking about it and members of my team were incredibly skeptical. I think now the excitement and the pipelines are filling, the backlogs and the sequencers are essentially running at capacity, and so the line of sight there. We're really looking to continue this partnership model. So as I mentioned, beyond the billion cell project, I think this initiative and back to this kind of core observation that while biological data is abundant, we lack the right kind of data, and looking to build partnerships to generate more of the right kind of data in the biological space for fundamental biological questions. In this case, I gave you an example of transcriptomics, but thinking much more broadly across new modalities and across other biological areas, but doing so in a way that really increases or maintains the pace of data generation such that it is closer to the speed that we're seeing in the frontier model space. And then the third point is one that's near and dear to me, which is I think as we start to think about AI models and as we start to push the limits of what we can generate data on, we can begin to think about new areas of biology that right now are not tractable. And even within the billion cell project, again, large data sets will get generated and models trained on it, but the questions that we're asking about cell type evolution, about how genes function across different cell types and perturbations, require a scale of experiment that previously would have been too long or too costly. And so part of this model is really rethinking how we can generate those data sets in speed and cost such that we can start to approach questions that right now just seem intractable. And so it's really trying to push forward the scientific doors and fields that perhaps many, including myself at various points, just did not feel that we could get to. Models then are a great way to generalize that knowledge and really expand how we are working on those fields. And then finally, a final slide, and something that is of course near and dear to our work, it has always been kind of at the bedrock, is our commitment to open science. So all of these data sets that we're generating are being supported via CZI as a philanthropy. Much like with Cell by Gene, the data sets themselves will be openly shared. They are being generated with the intention of modeling, and what that means really is that it's not just a large corpus of data, it also includes the infrastructure, the tooling, the data loaders, the tokenizers, other ways to enable researchers around the world to utilize those data sets for training new models, as well as a commitment to open sourcing and making available the models that we and others will train. Again, very much with the intention of pushing open these fields and both the models and the data serving as a catalyst to build collaboration and build communities at that intersection of AI and biology. And with that, I will thank all of you for your attention. I think I will hang out here. If anyone has any questions or if anything's burning, please come say hi and stay in touch. Thanks a lot for the time.